So in this video, we are going to talk about gluconeogenesis. So in my last video, I have already talked about the glycolysis, which is the reverse of gluconeogenesis, like the breakdown of the glucose. And in this video, we are going to talk about gluconeogenesis, which is the formation of glucose. So please make sure to like, share, and subscribe my channel. And to know more about glycolysis, please visit my last video. So, what is gluconeogenesis? So, gluconeogenesis is the synthesis of glucose from non-carbohydrate compounds. So, what are the non-carbohydrate compounds like pyruvate, lactate, amino acids? Because this reaction occurs when there is like a depletion of glucose, like during starvation. So, in that case, you need the non-carbohydrate compounds to be metabolized and form glucose, and it occurs mainly in the liver and to a lesser extent in renal cortex. Like glycolysis, it happens like in the cytoplasm of all the cells, but gluconeogenesis, it happens mainly in the liver. And where in liver? So the reaction partially occurs in the mitochondria and partially in cytoplasm, the opposite of the glycolysis, where all the reactions were occurring in the cytoplasm. And the major substrate, as I have already talked about, are the non-carbohydrate compound like pyruvate, lactate, glucogenic amino acids like glycine, serine, which can be converted to glucose, and then glycerol, which is released by lipolysis. So, when does this happen? So, as I have talked about during starvation, gluconeogenesis is important to maintain the blood glucose level because during starvation there is no source of energy by the diet by eating. So you have to metabolize the stores that are already present in the body, and then you maintain glucose by gluconeogenesis. So this is for the temporary starvation, and for prolonged starvation, this reaction is speeded up, and the major substrate that becomes is the protein catabolism, mainly the amino acids which can produce glucose with like glutamic acid, serine, and glycine. So the reaction that happens in the gluconeogenesis is, as we have already talked in glycolysis, it's the reverse of glycolysis because glycolysis is the breakdown of glucose and gluconeogenesis is the formation of glucose. And if you remember, in glycolysis we have talked about the reaction. There are three reactions which are irreversible, but the rest are reversible, like they can go in both the direction. But there are three irreversible reactions which can only occur in the forward direction, and the enzymes are hexokinase, which is the first step of the glycolysis, pyruvate fructose kinase, and pyruvate kinase, like the phosphofructokinase, uh, as we have already talked in the glycolysis reaction. So just to bypass these, and there are alternate enzymes which are available which can do the job of gluconeogenesis. So these are pyruvate carboxylase. I will talk in detail about this enzyme and how they function, like all the pyruvate 1,6 bisphosphatase, glucose 6 phosphatase. So I will talk in detail how they work. So if you remember this reaction, we have talked in the glycolysis where the glucose is broken down to two carbons of pyruvate. So this reaction, like from here to the glucose to pyruvate, is glycolysis, and the opposite direction from pyruvate to glucose is gluconeogenesis. So as you can see here, the major reactions are in reversible direction, which means that the same enzyme can perform both the function. So if you look here, from glucose to glucose 6 phosphatase, this reaction occurs only in one direction by hexokinases. So when we are going back from pyruvate to glucose, we need another enzyme which can perform this function, which is glucose 6 phosphatase, means it removes the phosphate groups and form glucose. And then the other step is the formation of fructose 1,6 bisphosphate by the enzyme phosphofructokinase, and this reaction also occurs in the one direction, which means then it becomes the rate limiting step as well. So to do the reversible reaction, there is another enzyme which is called the fructose 1,6-bisphosphatase. And then the rest of the reaction are in reversible order, like one enzyme can perform both the functions. And then is the last step where the phosphoenol pyruvate gets converted to pyruvate by pyruvate kinase. And this reaction is also irreversible. So to do this, there are two enzymes which are involved. So the pyruvate cannot be directly converted to phosphoenol pyruvate. It first gets converted to oxaloacetate, which is a 4-carbon compound, by adding a carbon group from the pyruvate carboxylase. And then from oxaloacetate, it gets converted to phosphoenol pyruvate by phosphoenol pyruvate carboxykinase, which removes the carbon group. So I will talk in detail about how these four enzymes works in gluconeogenesis, but they are not present in glycolysis. So we talked about the reverse, like when it happens from pyruvate. So from pyruvate to the oxaloacetate, pyruvate carboxylase work, and it converts the pyruvate to oxaloacetate with the use of ATP. And then in another step, like oxaloacetate gets converted to phosphoenol pyruvate by the action of GTP, which is another guanine, like adenosine and guanidine triphosphate, which again uses energy. So if you see this reaction from pyruvate to glyceraldehyde 3-phosphate, which is the pair phase during glycolysis, this reaction uses a lot of ATP. So this is energy consuming process. But because the maintaining the blood glucose level is very important to be used by the other sources, that's why this reaction occurs during the starvation. So first pyruvate get converted to oxaloacetate and then to phosphoenol pyruvate. And then if you remember, I have already talked to you in my last video of glycolysis, where you can see how these changes occur, like where phosphate group is added, where phosphorylation happens, and where there is isomerization. So you can watch that video, and then you see from phosphoenol pyruvate we go to phosphoglycerate, which releases a phosphate group, and then there is this isomerization from second carbon to third carbon, and then we use again energy to produce the 1,2-bis phosphoglycerate, like we add one phosphate group and then the dehydrogenase which form the glyceraldehyde 3-phosphate. So this reaction is the same as glycolysis but in the opposite direction, but this is the major reaction which then limit the rate of the reaction where it is going either in the forward direction or in the backward direction, and with the utilization of ATP. And then from glyceraldehyde 3-phosphate, so if you remember the glycolysis, we go in this direction, but in gluconeogenesis, the steps are opposite where glyceraldehyde 3-phosphate get converted or tautomerized to dihydroxyacetone phosphate, and then it forms the fructose 1,6-bisphosphate, which again to form this fructose 6-phosphate is an irreversible reaction. So we need another enzyme, fructose 1,6-bisphosphatase, and form this 6-phosphate, which releases one inorganic phosphate. And then again, this is a reversible reaction to form glucose 6-phosphate, and then from glucose 6-phosphate to glucose by the glucose 6-phosphatase and releases again the inorganic phosphate. So this reaction occur in the reversible reaction, and there are three major steps which are irreversible in this reaction, and this step utilizes energy. So now we know how this happens, glycolysis and gluconeogenesis. So now we are going to talk about what regulates the gluconeogenesis. So if you, so now we have talked about how this reaction occur. So if we have tons of acetyl coenzyme A, because this pyruvate can be formed by the acetyl coenzyme A, it occurs in this direction. But um, if we don't have the ADP, like the ADP, then this reaction inhibits because we need ATP for this reaction. And if we have ADP, then this reaction is inhibited because we get lots of ADP from this reaction. And similarly, if we have citrate, because citrate can be converted to oxaloacetate, and then this reaction occurs in the gluconeogenesis pathway. But again, AMP, because AMP means like we need ATP, which can be converted to ATP. But by using ATP in this reaction, because we require ATP for this reaction to occur, so AMP act as a negative feedback mechanism. And then fructose 2,6-bisphosphate. So this is 1,6-bisphosphatase, but we have fructose 2,6-bisphosphate, which also act as a negative feedback in this mechanism. And if we see on the other side of glycolysis, all this that is acting as a negative feedback here, they act as a positive feedback. So if we have tons of ATP, then this reaction will be inhibited because this reaction also generates ATP. Again, if we have 1,6-bisphosphate, then this reaction acts in the forward direction. But if we have ATP, alanine, which is um, mm, a glucogenic amino acid, then this reaction is also inhibited. So I hope it's clear how gluconeogenesis work and what pathways regulate this reaction and what are the compounds that can decide whether this reaction goes to form glucose or goes to form the pyruvate. So if you have any doubt, please let me know in the comment section and please like, share and subscribe my channel. Thank you.